Okay, so we're here today with Professor Lane Gilchrist of the CCNY Chemical Engineering Department. And it's September 20th, 2023. And Lane, we're very happy that you joined us today. And we'd like to find out more about your life, what types of things you're interested in, what kind of research you'll be doing. So,
well, thank you so much for taking the time to interview me, Stu. We really appreciate it. The department really appreciates your efforts. I started as a undergrad chemical engineer many moons ago at Louisiana State, and did my concentration in bioprocess engineering. But then I decided to go off to grad school and really work in the area of biophysical chemistry and magnetic resonance spectroscopy, and it was mentored along the way by really interesting people. Mary Barkley is a very prominent person in fluorescence spectroscopy, and then went on to University of California at Davis and worked on photosynthetic oxygen evolution, studying the enzymes in oxygen evolution that make all the oxygen on the planet Earth that come from spinach. And I had some of the funnest experience of my life going into the local grocery store in California and getting that wonderful California spinach by the bushel and grinding it up and making it into this green peanut butter like substance that would evolve oxygen if you shine light on it. And then when you could put it down at near 4.2 K in a magnetic field, shine microwaves on it through a waveguide and see the spin interactions of oxygen evolution and water molecules that are turned into oxygen, their signature on the catalytic site, which made of manganese. And for me, it was like
spinach was that interesting.
Yeah, I know. They thought I was crazy, but it felt to me almost like I felt at that time in Dave Britt's lab like I was a Magellan of the nano scale and the nanochemistry of photosynthesis. It was a fantastic experience. And that led me to then go on to work on magnetic resonance. Another great mentor I had, Ann McDermott, over in the chemistry department at Columbia University, studying various membrane protein complexes of different types, from the acetylcholine receptor to bacterodopsin, doing solids NMR. And then I, through various people that I met, including Dave Rumshitsky that I met at a conference, found out about this position over here, and we barely had anyone in the department that did much bioengineering at that point in time. So I came over and started my lab and wanted to basically look at really interesting systems on surfaces, go within the theme of interfacial phenomena within the program, and study membrane proteins on surfaces and build things out of them, engineered membrane proteins. And that led us over a many-year stretch to try and find interesting ones to work on, and I did a sabbatical back in 2008. I have Memorial Sloan Kettering with Yiming Li, who's a fantastic researcher in this enzyme called gamma secretase and its chemical biology. So we've been collaborating on that, and that's led us to this point where we are now. We've been studying that enzyme that's found in the human body and the brain and in cancer, and it's very relevant to signal transduction in cancer and metatasizing of tumors, and can be shut down by gamma secretase inhibitors and lead to therapies for cancer. So we go at it with different types of enzyme assays, largely 3D microscopy using fluorescence and super resolution fluorescence microscopy, but we have a whole side of the lab been working on, which is trying to grow various types of interesting microorganisms and gain different species, extremophiles, ones that grow in super high salt, and take in hydrogen and CO2 and then convert it into methane. This is a species called methanolholobium that grows at some ridiculous amount of salt, is like 10 molar salt concentration practically. And just trying to grow really exotic microorganisms in bioreactors, and train the undergrads to do bioprocess engineering, which is one of my favorite classes to teach. I love to teach, and I feel very blessed to be able to teach these classes, and it's a real privilege to teach some of the classes I get to teach at this institution. Really enjoyed that part. So that's kind of my long-winded explanation of that.
Okay, so if someone was, if I were a student that were considering coming to CCNY and sort of had you on my list of potential PhD advisors, what type of project what would I get into doing now? What would my life look like?
Well, one thing we've been doing, we've been looking at racial disparities in triple-negative breast cancer, but in the context of this enzyme gamma secretase, which is a protease, which makes fragments that are then sent to the nucleus to turn on the transition for the cells from static to then migrating into the body. And we've been looking at that process and the enzymes behind it and growing cancer cells and then getting them to shed their plasma membrane that contains endogenous gamma secretase. And studying the biophysics of it, the kinetics, and trying to understand the way it interacts with lipids and lipid bilayers and its lipid phase behavior, which is a really important thing we've been working on. And my students get training in a lot of different types of biological things, from growing cells to isolating lipids to making to studying lipid bilayers with and without proteins in AFM, confocal microscopy, superresolution microscopy. And we're aiming towards doing some NMR and going back to some NMR as well, some solids NMR. So, and my students end up, I've had my PhD students, there are quite a few that are out actively in industry at places like Janssen, Bristol Myers Squibb, let's see, there's some at Regeneron that we know of lately we've talked to, in Merck, that have done some training in my lab and gone out and are and have been really active in bioprocess engineering and formulations. So
Sure, chemical
to
engineering applied to bioprocess stuff.
So we've been lucky to have a lot of our students, undergrads and graduate students, in multiple groups in the department, go off to work with big pharma, doing bioprocess engineering style projects. In particular, growing proteins or other therapeutics, but also the formulation side, where they're figuring out how to formulate them into something that has a long shelf life. And so we have a couple of active people working on that, including Maldarelli and Two's groups on that side in our department. So we've been having real active placement of our PhD students and undergrads out in big pharma lately.
Okay, sounds good. So if I were a student that was thinking of coming to CCNY and maybe considering signing on to do this type of work, what type of advice would you like to give me?
Well, that's a very interesting question. I think I am the greatest proponent of going to graduate school. We're all kind of biased towards it that do academic research, but I just pour on the enthusiasm from the very beginning about how interesting and fun it is to delve into the parts of the books you never delved into. When you're an undergrad, you have this book that has 30 chapters, but you don't cover the last 15 chapters. You start to really get into the details and really go under the hood at what you learned about as an undergrad and the details behind it, and then start to apply those and then engage in a research problem that more often than not becomes very difficult, and you have a giant hurdle to go over in your PhD. Right. And by the time you get to the end of your PhD, you feel like you've really done something, and you feel very accomplished, and you're ready to take on the world. And but it's so wonderful to be able to direct your energy to something really exciting as a graduate student. It's magical. I tell the students sometimes that I sometimes wish I was back in graduate school.
Yeah.
And I mean,
it makes the next 40 years of your life. It charts that.
And I would say you have to choose carefully who would mentor you and what they're doing and how it's going to train you with various techniques you might go off and use. Right. I think that relationship's hugely important. I've been very blessed with my mentors that I've had. I'm very thankful for the ones that have mentored me.
Yeah.
And I sometimes joke that maybe I peaked in grad school, but
I don't think so.
but I sometimes miss grad school. But anyway, yes.
Yeah.
You have to cut this part. Cut.
So, okay, so on a final note, do you have any other comments or thoughts you'd like to share that we haven't touched so far?
I would say to those of you that are thinking about going to grad school and where to choose to go to grad school, I think that one of the things that is the hallmark of this institution is, at this point in particular, is that the research facilities that we now have in place between the labs on CCNY campus and the Advanced Science Research Center are absolutely and totally top-notch. And there is nothing holding you back, minus what you can think up to experiment on. So we have zero limitations, and you have the access to the greatest experimental tools that we affectionately call toys for graduate students at this institution. And I'm really excited to have been part of building those up over many years where we didn't have much infrastructure. Now we have world-class scientific infrastructure and instrumentation, and I use every excuse I can to go and train the students so I can run the instrumentation myself.
Okay, we like the enthusiasm. So thank you so much for joining us today, and thanks for your comments and your wisdom. Appreciate it.
Thank you very much, Stu.